Look at chapter number 1, and this is a really nice introduction into this class. We're going to talk about a really wide span of things that you are going to learn the entire semester long. So starting off, we'll kind of see what the term microbiology means, what exactly we're going to look at. So as far as the term biology, what do you study when you are studying biology? Life, yes. Bio means life, ology means the study of. In this class, it's titled Microbiology. What does micro mean? Small. Yeah, we're going to study these little itty bitty tiny microscopic organisms. These organisms we cannot see with the naked eye. Up here I have, it studies these microorganisms, can be easy, can be difficult too. Why is it easy? Because bacteria are pretty easy to grow in the lab. It doesn't take much. Think about what we did on Tuesday. So I had that plate, right? I passed it around the room, everyone touched it, tossed in the incubator. I took a look at it this morning. There's all kinds of bacteria growing on there. How much effort did that take? Nothing. As long as you have the right media, we had the right plate, all you had to do was touch it, put the bacteria on it, and they started to grow like mad. Okay, so pretty easy, as long as you know a little bit about bacteria and right type of media to use. Can be difficult too. Why is it so hard? Well, can you actually see the bacteria that are crawling on your pencil right now, or all over your tabletop, or on your skin? You can't see them, right? We can't see these organisms. They are too small to be seen with the naked eye. So it's difficult because we don't really know where they're at. We can't see them unless we have some type of a tool like a microscope. Or if we grow them up in mass numbers, like colonies on a plate or in a broth tube, then you can see just the clusters and clusters and clusters of cells. As far as other terms, microorganisms and microbes, that's typically what I'll use when I talk about these bacteria and fungi and parasites and so on. You'll also see bugs and germs. Maybe your textbook doesn't really use them very often. They're not proper terms. Because when you hear bugs and germs, does that make you think positively or negatively about bacteria? Negatively. Well, I can tell you that there's like this many bacteria that exist. There's this many that can actually hurt you. So that means most of the bacteria in existence are actually good bacteria. They do a lot of beneficial things for you and I, for our environment, and so on. So those are kind of unfair terms that are used to describe some of these microorganisms. Okay, a little bit of history. So just a brief look at evolution. We're not going to go too much into evolution, but here we can see a nice statistic. So we're looking at when bacteria first appeared. So here we're looking at about 3.5 billion years ago. We can look at this table down here. I'll just zoom in on this table a little bit. So here we can see probable origin of the Earth, really long time ago, right? About here, these ancestral cells, so your first bacteria, we're looking at between 3 billion, 4 billion years ago. Within here, what do we see? We see bacteria, eventually archaea, and then something known as eukaryotes. So looking at these bacteria, about 3.5 billion years ago. If we scroll up here, look at us. Here's humans. All the way to the right, we're at the present time. So these bacteria have been around way long before we have. Should we try to eliminate all the bacteria in our environment? No. Could we do it even if we tried? No. Okay, so they've been around a long time. They figured out a few things about how to exist, okay, and just kind of persist on this planet. So definitely they are beneficial to all of us. Here we can see a little bit about evolution. We'll get into that in a later chapter. So a few details. Zoom in back here, and then over here. Okay, a few terms that I want to put for you up on the board. These terms. Have any of you ever heard eukaryote or prokaryote before? Yeah. You've heard those before. Okay, so we're talking about different types of cells So if you're looking at bacteria, you can say bacteria are, prokaryotes, they're prokaryotic cells. Eukaryotic, that would be like looking at our cells, for example, fungal cells, plant cells, those are all eukaryotic. When you look at prokaryotes, so I'll say, just sum it up and say proks, this is going to equal bacteria and what's known as these archaea. Now archaea, they derive their name from being ancient. They're very similar to bacteria, okay? At least that's what we thought. Doing a lot of genetic studies recently, we see that they are indeed quite different. So we used to just talk about eukaryotes and prokaryotes. Now what we're seeing every single time this microbiology book gets updated, what I'm noticing is they're taking the two prokaryotic types, bacteria and archaea, and they're separating them. So pretty soon they're going to eliminate this and just talk about bacteria versus archaea. Big difference between the two. Bacteria are your standard run-of-the-mill bacteria. E. coli, Staphylococcus aureus, Clostridium difficile. Okay? These are your standard bacteria. Your archaea, I'll say odd. I'll also say extreme. They're ancient. They've been around for a very long time. They live in odd and extreme environments. So if I were to ask you, do you think there's any bacteria, any of these prokaryotes that live way down in layers and layers and layers of ice down in the Arctic? No, there's going to be bacteria that live way down there in all those deep layers of ice. Sure, there definitely is. The archaea. What about within a volcano? Wouldn't that completely destroy bacteria? I think so, but archaea have figured out a way to adapt. What about in a super salty environment? If you were to put any bacteria like E. coli in a very salty environment, okay, it's going to completely dehydrate the bacteria and destroy it. However, archaea, they have adapted to live in these super salty environments. They actually use the salt for their metabolism. So they almost need to have that salt. So these guys have figured out ways, they have special adaptations within their cell to allow them to live in these really weird environments. So that's a little look at the difference between these two. What I need to talk about now is the difference between eukaryotes and prokaryotes overall. So let's do that. Another eraser. All right, so eukaryote, the EU, this means true, kary means nucleus. That's the biggest clue as one of the main differences between these euks and these prokaryotes. Eukaryotes, they have a true nucleus. I will put in parentheses, membrane bound. So they have a membrane-bound true nucleus. If you think about our nucleus, we have two membranes. It's a double membrane structure referred to as the nuclear envelope. So eukaryotes have a true nucleus. They also have, so they have a true nucleus and they also have membrane-bound organelles. By a membrane-bound organelle, we are referring to, let's see, the ER, the Golgi apparatus. What's the energy powerhouse of the cell? Mitochondria. That's responsible for making ATP. Okay, so they have these membrane-bound organelles. Now, if we look at prokaryotes, what do prokaryotes have? I'll just write out the whole term. Okay, so prokaryote. Pro means before. Kary means nucleus. So these guys, they do not have a membrane-bound nucleus. Okay, so I'll just say no nucleus. They do have a nucleoid region It's just where their genetic material is found, but it's not encased in any type of a membrane. It's just kind of freely floating within the cell. Okay, I use that term lightly, but there is no membrane around it. So no distinct nucleus. Also no, I'll just say MB organelles. No membrane-bound organelles. So that energy powerhouse, that mitochondria, how in the world do they take different energy sources and convert it into their ATP? How do they do this? How do they undergo their metabolism if they do not have mitochondria? Well, they've been around for 3.5 billion years. You can bet that they've figured out a way to do this. They have a lot of special adaptations that they can do right in their cytoplasm or across their cell boundaries. Okay, so they can still do a number of the same processes that eukaryotes can do. They just don't have those specialized organelles. Do you have a question?
Oh yeah, I'm just wondering in the notes, what does MOs mean?
Microorganisms.
Okay.
So yeah, if you see that, it's just an abbreviation for microorganism. One more thing I want to mention, eukaryotes. They're typically larger if you were to look at the cells, and I would say larger by a factor of 10 on average. And then prokaryotes, of course, these are smaller. Lab number 6, which we'll finish up next week, it'll be due next week as well on Tuesday. That one question that I mentioned, you might need to do a little bit of research in the textbook. One of the things they ask you is differentiate eukaryotes and bacteria based on size. Here you go. Eukaryotes are larger, prokaryotes are smaller. Okay, so questions on differences between these. Awesome. So here you can see a little wrap up of what we just went through, prokaryotes versus eukaryotes. Here you can see some images of some cellular differences. So if we look at the prokaryotes, which contain bacteria and archaea, here we can see a very simplistic cellular structure. So you do see the chromosome here, but there is no distinct nucleus, like what you can see over here in eukaryotes. Also over here, we don't see any of those organelles that you would see over here with a eukaryotic cell. You do see ribosomes. Does anybody remember the function of a ribosome? Yes.
Something with protein?
Yes. Protein synthesis. Okay, so they are going to synthesize proteins. However, ribosomes, they are not a membrane-bound organelle. There is no membrane around them. So, yes, prokaryotes and eukaryotes both have them. And then, of course, taking a look at these cellular boundaries, we can see eukaryotes have a cell membrane. Prokaryotes, there is also a cell membrane, but additionally, there's a cell wall. If you look at our cells, we do not have a cell wall. We have a cell membrane, phospholipid bilayer cellular membrane. Prokaryotes also have a phospholipid bilayer cellular membrane, but additionally they have a cell wall, and then some also have an additional outer membrane. So they have a variety of barriers around their cell. If you look at eukaryotes, okay, so if you're looking typically at the cell membrane, you'll see some eukaryotes, such as plants, for example, having a cell wall and it just has some different chemicals in it compared to a bacterial cell wall. But if you're looking at a human cell, of course, we're looking at just a cell membrane. And then we have the viruses. We're going to spend an entire chapter talking about viruses. And the big thing at this point to understand about a virus is that it is not a cell. There's absolutely nothing cellular about a virus particle. If you were to call it an organism, a cell, you would be completely incorrect. It is not cellular. It is not a living organism. It is simply a particle. All that a virus is genetic material. It could be DNA. It could be RNA. Genetic material wrapped up in a protein coat. That's a virus particle. Very, very simple, yet very, very difficult to deal with if you're looking at an infection aspect. Okay, and we'll see why a little bit later in the chapter, or in the semester. Okay, so if I were to ask you if a virus is a prokaryote or a eukaryote, it would actually be neither. Okay, it's not prokaryotic, it is not eukaryotic, it is not cellular in any way. Virus particles technically are not living things, they're not living organisms. There's two things which are requirements in order for something to be a living thing. One of them is called the cellular theory. You have to be made up of at least one cell. Bacteria are unicellular. Awesome. We're made up of trillions of cells. Okay, so we are living organisms. A virus is not a cell, so it doesn't fit that bill. The other thing is something called autopoiesis. That means you're able to undergo self-metabolism and so on. These guys, they cannot do anything on their own. They cannot undergo their own metabolism. Okay? They cannot generate ATP, nothing. The only way that any of that would get done is if they were inside of a host cell. Essentially, you can think about them as little genetic hijackers because they will bind to a cell. There's different methods on how this works, but ultimately the genetic material will take over the genetic material in the host cell, and then that host cell now becomes property of the virus. And the virus essentially uses the host cell as a virus-making factory. Okay, so the host cell no longer belongs to that host. But a virus cannot do any of that on its own. It needs a host cell. Also, viruses much, much smaller than bacteria. We would never be able to see a viral particle on the microscopes we have in class. They're too tiny. Lots more to come with viruses as we move through this course. Here we can see exactly what we're studying in this class. So definitely viruses. So here's an image. Okay, so this is showing the herpes simplex virus. A few viral particles here. Bacteria, of course. Viruses we'll talk about in chapter number 6. Bacteria we'll talk about prokaryotes in chapter 4. That'll be next week. And then these three categories here, protozoa. Okay, so these protozoans, these are eukaryotic, unicellular organisms. We'll talk about them in chapter 5. Fungus. Okay, so here you're looking at fungi, you're looking at mold, also eukaryotic. We'll talk about them in chapter 5 as well. Does anybody know what a helminth is? I've heard that term helminth before. What if you look at this picture? What is that thing? It's a little worm, yeah. Okay, so helminths are parasitic worms. They're eukaryotic, of course. They are animals. Can you see helminths? Could you see a roundworm or a tapeworm with the naked eye? Sure you can. These tapeworms, they can get quite large. Okay, so in A&P, we've taken them out of the stomach of cats, okay, and they've been quite large. You're looking at basically like a foot in length. Humans have been known to have tapeworms 60, 70 feet in length, which is crazy. Okay, why do we study them in this class if they're animals and you can see them with the naked eye? What about them is infectious? What are the chances of you coming in contact with a 10-foot tapeworm? Not really likely. How would you get infected if you're looking at a tapeworm infection? What would you ingest? The eggs, and the eggs are microscopic. Okay, so that's what we're looking at in this class. We'll talk about a few specific worm infections, that'll be in chapter 5, but the reason we study them is because the eggs are microscopic and the eggs are what's infectious. You cannot see those with the naked eye. And then here, we can just see the size of things. So what we're taking a look at, this gigantic pink thing in the background, here you're looking at the head of that helminth. Okay, so right here, it mentions it's visible to the naked eye, of course. Human hair. Obviously, that's only visible to the naked eye. Protozoan, fungus, red blood cells. We're getting smaller and smaller and smaller. Right here, we can see the fungus, this blue guy in the background, in relationship to a bacteria. And then down here, we can see the size of a virus. So these viral particles, think about, this, if you saw bacteria on your slide on Tuesday, think about how tiny they were in that microscope. And then think about how little this viral particle is compared to that bacteria. No way we can see that in the microscopes that we have. So extremely, extremely small. Oh, that word in blue, that was in the title of one of the labs that we did on Tuesday. And what does that mean again? Ubiquitous?
Everywhere.
Everywhere. So we see that these microorganisms, these bacteria, they are indeed everywhere. So we'll check those results next week. But again, they do so much good. They're very beneficial to our environment, to other organisms in our environment, to us. Up here I have a couple of examples of how they help to keep the earth in balance. One of them is photosynthesis. We'll talk about the equation for photosynthesis and the equation for aerobic respiration and how they relate to each other in regards to ATP and so on a little bit later. But for now, if I were to ask any of you guys, which organisms out in our environment are responsible for photosynthesis, what would you tell me? Plants. Number one answer, plants. Okay, so now if I tell you that bacteria and algae are responsible for over 70% of all of the photosynthesis on our planet, that is crazy. Okay, so plants are only responsible for much, much less than the bacteria and the algae. Okay, so you're looking at no more than 30% for all those plants. So yes, the bacteria are doing a great deal of work in algae as far as being responsible for over 70% of the Earth's photosynthesis and oxygen production. So that's kind of an alarming stat. And then also decomposition. So this is going to be recycling nutrients from dead, decomposing organic matter. Okay, so these microorganisms are going to recycle these nutrients, put them back into the earth, and so on, kind of keep that circle of life going on. So they do a lot of good, regulating our air temperature, providing nutrients to plants, and so on. A little look at human use of microorganisms. So we are utilizing these microorganisms all of the time. If you like to have an alcoholic beverage every now and then, so looking at wine or looking at beer, yes, you are looking at these microorganisms helping to produce that product. Actually, in this class, we are going to take some grape juice and some yeast, and we're going to let the yeast eat up all those sugars in that grape juice and create some alcohol. So we're going to make a wine-based product, which is kind of cool. You just obviously can't drink it, but it's actually going to smell like wine. Okay, so we're going to see how these microorganisms can do something like this. Also, the production of different types of cheeses. I don't know who the first person was that was like, I'm going to try this moldy cheese and see if it's any good. But if you like blue cheese, hey, I mean, someone had to try it first and be like, this is awesome. You know, some of these, these microbes help to create this product. Proprioni bacteria helping to create Swiss cheese, produce the gas that's responsible for the holes in Swiss cheese. So yeah, they do a lot of good. Looking at baked goods. Of course, helping the bread to rise, looking at yeast. Antibiotics. If you think back in time, if you look at a little bit of history, what they used to do back in the day was if someone had a cut, some type of an injury, they would take a moldy loaf of bread and they would slap it on the wound. Again, who thought of this? I don't know, but it was brilliant, right? Because if we think about antibiotics and how they're made, they're byproducts from bacteria, or they're byproducts from some type of mold. Penicillin is made from the penicillium mold. Streptomycin is made from Streptomyces. Okay, so they're made from other organisms. So yes, taking that moldy loaf of bread and slapping on a wound, there's definitely benefit from that. And we were able to create all these antibiotics as a result. If you look at the term antibiotics, It has the term bio right in it because that means it was generated from some type of a living organism. These living organisms, like the penicillium mold, they have these factors that kill other organisms just because out in nature, they don't want any other organisms invading their space, okay, taking over their territory. So they give off these properties that kill other organisms. So scientists kind of caught on to that and just harvested those byproducts and developed them into these drugs that we use today. Big topic that we'll talk about this semester is going to be overuse of antibiotics and also drug-resistant microbes. Huge, huge topic. And we'll get into that about the second half of the semester. Genetic engineering, controversial topic. Okay, so you're utilizing these microbes and engineering them in different ways. Bioremediation, here you're looking at using these microbes to clean up something toxic, maybe like an oil spill. And that's what's going on in this picture down here. Okay, so these microbes can digest this oil and help to clean up the environment, okay, and make this process much, much easier. And if you think about a septic tank, you can look at microbes helping to clean up sewage. They also help to clean up water, so they can do a lot of good. Here's the scary part. Okay, so again, this many microbes exist, this many can hurt you. So here's a few details about these scary microbes. We're looking at 2,000 of them, which would be considered pathogens. Pathogen, patho refers to disease, gen means to generate. So pathogens are disease-generating organisms. They're responsible for infection and disease. 10 billion new infections each year. It's more than the population on this planet because oftentimes individuals come down with multiple infections per year. There's a table up here. This table is taking a look at top causes of death, all diseases. So if you look at the United States, okay, right here in pink, I put a little arrow right here. This is going to be the only category, so number 8, we look at influenza and pneumonia, that is related to microbes. Okay. Everything else on this list, okay, we're looking at lifestyle, genetics, and so on. Over here, if we look worldwide, diarrheal diseases, number 5 worldwide. That's kind of alarming. Excuse me, HIV/AIDS, number 6 worldwide. Okay, neither one of these are on our list. TB, number 8, again worldwide. Here we're looking at those lower respiratory infections as number 3 worldwide. So definitely respiratory infections, okay, extremely, extremely prevalent. But some of these other ones, why do you think these are seen worldwide and not so much looking at the United States? What would be a reason for that? Treatment availability, healthcare system, economy, living conditions as well. There's a lot of different factors. So if you look at what we have in this country, definitely we have a good healthcare system and so on. We are seeing heart disease number 1 worldwide, of course in the United States. We're also seeing accidents up here. Okay, where we don't see that over here. We do see road traffic accidents. If we scroll down here, there's another table. This table is taking a look at countries based on income. So you see low-income countries, these communicable diseases, okay, look at how high. Compared to, as you increase income, okay, you're not seeing this huge number. It completely drops down. And then meanwhile, over here, chronic diseases, okay, very high. It drops a little bit, and then high-income countries, chronic diseases are high again. Again, a lot of it has to do with lifestyle. Okay, these high-income countries. So it's kind of an alarming table to look at. Malaria stats, okay, kills 1 million people every single year. Drug-resistant microbes. Okay, hot topic. You know, if you were to look, I don't know, maybe like 100 years ago, there was a statistic that 1 out of every 3 children would die, okay, before the age of, I think it's like 10, with some type of an infectious disease. Okay, 1 out of every 3. That is insane. That was before antibiotics. And then all of a sudden, antibiotics came around, and it was a miracle. It was miracle drugs, these antibiotics. And they were curing, you know, all these children with these infectious diseases, and it was fantastic. And that continued on, and then we started to see a little problem with these antibiotics, right? All of a sudden, some of them weren't working very well. Well, this issue, I mean, technically it really started in like, you know, 70s or 80s. But when did we hear about, you know, these drug-resistant microbes? Really didn't hear about them very, very commonly until the last, you know, decade or so. Why is that? If they've been around, like, why have we heard about them? Well, back in the day, you know, if you're looking back in the day, like 70s or 80s, you know, if there was some type of, you know, drug resistance, well, we'll just give drug B, and then drug B did the job. Awesome. If something was, you know, resistant to drug B, we'll just give drug C. Awesome. Not that big of a deal, doesn't need to be in the news because there was always backups. Nowadays, all right, drug A doesn't work, let's try drug B. That doesn't work either. What about C? That doesn't work. Let's try drug O. That doesn't work. What about drug T? That doesn't work. Okay, we have nothing. Okay, so now that's the problem. We don't have these backups. All of a sudden there's no antibiotics that are useful against that particular organism. So it's just kind of getting worse and worse and worse. There's a lot of politics involved with it and so on too, and we'll talk about that a little bit later in the semester. Let's do a concept check. So let's review a few things we talked about. So what types of microbes or what types of organisms are considered microbes? So let's hear a few. So anything microscopic, so such as, what are we growing in the lab? Bacteria, good. We also have algae, we also have fungi, we also have protozoa. Okay, these are all microscopic organisms. What types of cells can viruses infect? I didn't talk about this one, so now's a good opportunity. So, can we be infected by a virus?
Yeah.
Silly question, of course we can be infected by a virus. Every one of us in this room has had a cold at some point. That's the rhinovirus. Okay, that's a virus. What about that tree outside?
No.
Actually, yeah, definitely. There are some viruses that will infect plants. Okay, so plants are susceptible. What about bacteria? Yeah, bacteria are susceptible too. There are viruses out there that target bacteria. In fact, something kind of interesting, bacteria, if they have a viral infection, this is going to sound crazy, but if bacteria has a viral infection, that bacterial strain is much more dangerous to you or I. Because they have that viral infection, they may have the ability to produce some type of a toxin that they would not normally be able to produce. Okay, so if they didn't have that viral infection, no toxin, that bacteria can't make us sick. If they do have that viral infection, now they have that virus genetic material incorporated with them, they can make a toxin, they can now make us sick. Okay, so yes. So the answer to this, what types of cells can viruses infect? Everything. Any living organism is susceptible to some type of a viral infection. Prokaryotes versus eukaryotes, you should be able to tell me differences between those. True or false, microscopic organisms produce more oxygen than plants? Absolutely true. What was the percentage?
70.
Yep, 70. True or false, all infectious diseases are contagious?
No.
Let's talk about this one too. We need to talk about a disease versus an infectious disease. If we talk about heart disease, is heart disease an infectious disease? No, it's not. Is cancer infectious? No, it's not. Is malaria infectious? Yes. It is. Is C. diff infectious?
Yes.
Yeah, it is. Okay, so let's do this. I'll write on the board, infectious. What causes C. diff?
Too antibiotics.
Okay, so all those antibiotics kill the good guys, okay, so the good flora, and then those C. diff spores are going to have some opportunity to emerge into bacteria, and then you're looking at that bacterial infection. So C. diff responsible, from the bacteria. What is responsible for AIDS? HIV, which is a virus. Okay, so we see something caused by bacteria, we see something caused by virus. What is ringworm? Fungus. Okay, so we're looking at a fungal infection. Okay, so those are all microorganisms. A virus technically isn't an organism, yet it's small and infectious. So infectious diseases, I'll just say MOs. Okay. Infectious diseases are caused by some type of a microorganism or a virus. That differentiates an infectious disease from any other disease. Now, question is this: are all infectious diseases contagious? Can you spread all of them to another person? If you had an E. coli infection, could you spread that to another individual? Mm-hmm. Sure. What about MRSA?
Yeah.
Sure you can. What about if you had the flu? Mm-hmm. Sure. Okay, so you get the influenza virus. What about, how do you get Lyme disease?
Ticks.
A tick. What about the tick though? Is it just that type of tick that gives you that infection? Okay, it's a specific type of a tick, so it's going to be a deer tick. So there's a microbe in it. That's the big key. So if you get bit by a tick, okay, well, looking at Lyme disease, it would have to be a specific type of a tick. Not only that, but that tick would have to be carrying a certain bacteria. It's called Borrelia burgdorferi. It's carrying that bacteria. When the tick bites, it's going to inject that bacteria into your body. So Lyme disease is caused by a bacteria. You just get it from a tick. Okay, that's the source. So it's caused by a bacteria. Now is Lyme disease. So Lyme disease fits this bill, right? It's infectious because it's caused by bacteria. Is Lyme disease contagious?
No.
No, it's not contagious. You're not going to pass Lyme disease to your spouse or to your children or to your friends. So now let's revisit this question. All infectious diseases are contagious? That's false. Okay, they are not. Lyme disease, perfect example. Okay, it's caused by a microorganism. It is not contagious. It's not communicable. You cannot pass it to another individual. Let's talk about some history. And with this part, I will be bringing up a few different names of individuals, and I'll expect that you link the name of the individual to what they did, what they're responsible for in the world of microbiology. So here, we can see that, oftentimes foods were spoiled, and you guys have this. If you forget you had a loaf of bread, in the cabinet, and you're like, oh, there's that loaf of bread. It was hidden behind a few plates, and you look at it. So I got some mold growing on it. Are you going to eat it? No. Okay, I wouldn't. Okay, so definitely is not going to enhance flavor in any way, but like we talked about with blue cheese, here you're looking at the mold actually enhancing flavor. Sometimes the enhanced flavor cause no harm. Other times, it might be dangerous. And they really didn't know what it was. Like, what is this on the bread? What is this on the cheese? They couldn't see these microbes until we had our first gentleman right here by the name of Robert Hooke. He was the one who was able to observe these mold colonies. If you look very, very closely at some mold on bread. And I'm already growing some moldy bread at home that I'll bring in for our mold lab that we'll do in a couple of weeks. Everyone in this room will also receive a plate, which I'll talk about specific media in the next chapter, but you'll take it home and just expose it to the air, and you'll be surprised at all the mold which is going to grow on that plate just from the air in your home or workplace, wherever you want to take it. Just leave it in the hallway if you want to. And if you were to look at the mold up close, what you are going to see are these little spore-bearing sacs. Okay, and these little spore-bearing sacs contain hundreds and hundreds of little mold spores. And all that it takes to start growing mold is one little spore to land on the bread. A lot of times by the time you purchase the bread, it's already, there's a couple on there. But they're going to spend some time germinating throughout the bread, just exposing them to the air. You're going to have some land. And eventually they kind of pop up. Hundreds are in here. Eventually the spore-bearing sac bursts. Hundreds get released, and they're going to settle on another portion of the bread. Germinate throughout. More of these pop up. They're all going to burst. More spores come out. Purpose of mold spores is going to be reproduction. You might want to jot that in your notes. Mold spores are for reproduction. That is the purpose of them. Just to make more and more and more and more mold. Once it starts, it happens very, very quickly. It just takes a little bit of time for it to get going. But Robert Hooke was the one who first observed these spores. Then we have the guy with the really long name. Okay, the guy with the really long name developed something that's going to allow us to see really small things, which would be the microscope. Antony van Leeuwenhoek was the gentleman's name. He wasn't even a scientist. Well, I guess he was by nature, in the sense that all scientists are just, curious and just want to know more about everything. And yeah, he was very, very curious. He was actually a tailor, worked in a thread shop, and he devised these lenses so he can see the thread count better on the articles of clothing that he was working with. He's like, okay, I can see the thread count better. What else can I see with this lens? He was very, very curious. Let's see the rainwater that landed on his windowsill. So he took some rainwater, took his lens, and he's like, wow, the water looks alive. If I, kind of see it magnified, it looks like there's little things in there. They're like little tiny animals, but they're like molecules. So that's the word he developed, animalcules. He's like, wow, they're running around in this water. And he's like, what about this stuff between my teeth? This gook between my teeth, this plaque. So he scrapes them off. He used that lens, and he's like, wow, there's even more of these little animalcules between my teeth. And then he decided, does anyone else have these little animalcules between their teeth? So then he went around and he scraped some other people's teeth. He's like, wow, everyone's got these little animalcules. So eventually, this little lens, he developed this contraption to house the lens, and this was your first primitive microscope. So the guy with the really long name developed something to be able to see things really small. Okay, kind of an easy way to remember it. And it was very simple. There's no light source, don't need to plug it in. Specimen holder at the end of the screw. There's a focusing screw to bring it closer or further away from the lens. That is it. Okay, so that's his primitive microscope. Here you can see it in use. Very, very simple. This is taking a look if you were to scrape up some plaque from your teeth. That is what you would see. If you have all these microorganisms in a very complex community such as this, and this is what you would see if you were to look at, like catheters are known to have, if they're not, treated properly, have biofilms growing on them, shower stalls, pool liners, and so on. Very, very difficult to get rid of once these biofilms start growing. It's just complex community of different organisms giving off byproducts that attracts new organisms, and they all start to build. And like plaque on your teeth, for example, once this biofilm starts, it happens very fast, and it becomes a very solidified structure and hard to remove. So the dentist comes in with little scrapey tools, like just digging away, at your teeth. It's not like a soft community. They're very difficult to remove. Okay. Here we get to one of our most important scientists. But before that, what was perceived back in the day was, so-and-so that lives down the street, they're a bad person, they're naughty, they're a sinner, that's why they got sick. They're kind of thinking more along the lines of this karma thing, right? These bad people are the ones that got sick. And this gentleman by the name of Louis Pasteur, right here, Louis Pasteur, he's like, no, that can't be right. They're getting sick from something. This growth, this bacteria, this is coming from something. Life is not just going to spontaneously appear without coming from a source. So what they just thought, no, they're just, bad person, that's why they got sick. But what Louis Pasteur did was he disproved, so I'll write disproved spontaneous generation, which is the same thing as abiogenesis. Genesis refers to the creation. Bio is life. A in front of it means without, which is spontaneous generation, that life just spontaneously appears. Okay, so creation of life without any other life. He's like, no, that's not right. That can't be. So what he did was he proved biogenesis, which is life comes from life. That life appeared from some other life before it. Life comes from life. What he did was his famous swan neck experiment. Swan neck flask experiment. And right here, you can see 2 swan neck flasks. So he took those 2 flasks, and what he did was, I'll just draw a little picture on the board over here. So here's one, greatest artist, so just kind of bear with me, even though I'll have you guys draw all kinds of pictures all semester long. But, so I'm going to draw the swan neck flask. So there's a flask with a crazy swan neck on it. I'm just kind of drawing a question mark with a flask on the bottom. Okay, so there's 2 swan neck flasks. In them he had some broth. He heated up the broth inside. When you guys grabbed the nutrient broth on Tuesday and you grabbed your swab, were you able to see through that broth? It was clear. So it was sterile. There were no microbes in it. So he heated them up to kill anything that might be in there. So he's starting with this fresh broth, has no bacteria in it. And what he did, so this one I'm just going to write clear. Okay, so this flask was clear. This other flask was also clear, but what he did is he cut off the top. So he got rid of that swan neck. So as you can see right here on this picture, one of them, the neck was cut off. The other one, the neck was still there. So what happened is, he's like, all right, if life is going to happen in these flasks, it has to come from some other life. Right now, there's no way for life to get inside this flask. This one, life can get inside because I cut the entire top off. So I'll erase this. The top is gone. So what happened? There's bacteria in the air. And bacteria settled in, like the nutrients, started to grow. So on Tuesday, when you check those tubes, and if they look all cloudy, and trust me, there's some cloudy ones in there, there's all kinds of gook that's formed up on the top, a nice layer of scum. Yeah, so here we see this one became all cloudy. This one's cloudy. This one stayed clear. Because bacteria in the air, they don't have wings or little feet, so they're not gonna like fly up in here or crawl and make their way around. So this one stayed clear. This one became cloudy because life comes from life. This life came from life in the air. So that was Louis Pasteur. He did so many things. He created the rabies vaccine. Also, he's responsible for pasteurization. Okay, So very, very important microbiologist. I'll give you one more example of an experiment that helped to disprove this spontaneous generation. There's a few in your book. This one was done by someone named Francisco Reddy, R-E-D-I. So I'll say Reddy's meat experiment. I think I have a little tribute to him up here. So Francisco Reddy. Francisco Reddy's meat experiment. What he did, he took 2 jars, jar 1 and jar 2. He put some meat in jar 1, he put some meat in jar 2. So right now I have 2 jars, each has meat. People would say, all right, the meat I bought, the other day at the market, bad because it produced maggots. He's like, no way can your meat make maggots. Those maggots have to come from some type of life. Where do maggots come from? Flies laying their eggs. So he's like, all right, let's see if I can show these people that the meat's not going to make these maggots. So with this one, he left it uncovered. The other jar of meat, he put a cover on. So now, what happened are these flies, and an F with a circle is my drawing of a fly. Okay. So we have these little flies coming along. They can't get to the meat with the one with the cover. So this fly's trying, but it can't. Over here, these flies are landing, laying their eggs, and all of a sudden, we have maggots. This one, we had no maggots, because those flies could not get to that meat and lay their eggs. Okay, so again, this life, these maggots came from life. They came from these flies. So a couple scenarios, there's a few more in your book. Spores, huge, huge topic. Remember we talked about mold spores? What was the purpose of a mold spore? Reproduction. Okay, mold spores are for reproduction. Mold spores are very easy to kill. They're not hard to kill. They're very easy. Up here, I have something referred to as a bacterial spore. Okay, bacterial spore, you can call it a bacterial spore, you can call it an endospore. What does endo mean?
Inside.
Inside. So an endospore is a spore found inside a bacterial cell. When you're looking at fungal spores, they're not endospores. Endospores are only for bacteria. Endospores are ridiculously difficult to destroy. There was a gentleman by the name of John Tyndall. You can see his name up here. He's the one who first discovered that it's pretty easy to kill some bacteria, but then other bacteria, they're not dying. I can't kill them. Okay? I'll use the same treatment methods that I use on some bacteria, but these guys, these disinfectants are not killing them. A few days later, the bacteria are back. Why are they not dying? It's because they produce these spores. These spores allow that bacteria to be resistant to heat, be resistant to chemicals, be resistant to some forms of radiation. Okay, so these spores are very, very tough. They are not used by bacteria for reproduction. They are used for one thing, and that thing is survival. Bacterial spores are used for survival. If you have a spore-forming bacteria and you attempt to destroy it, what that bacteria is going to do is form that spore. That spore is going to persist. As soon as the conditions are no longer adverse, as soon as those conditions are nice again for the bacteria, it'll turn back, that spore will turn back into the active bacteria. That's what happens with C. diff. C. diff, Clostridium difficile, that's an anaerobic bacteria. It will grow in places of no oxygen. So, someone has a C. diff infection, they go to the bathroom. Okay, so then they're releasing a lot of this bacteria. However, there is oxygen around. These are, you're looking at spores now being present. These spores are so tough to kill. Okay, they're landing on things. Maybe they're on like a hospital curtain. Okay, so they're on something, and they're going to persist for months. Not much is going to kill these spores. All that it takes is someone to ingest these spores, wiping out the good guys, and then these spores are like, hey, we're in an anaerobic environment, there's no good bacteria, we're going to break into the C. diff, go back to the bacteria and initiate the infection. You see the same thing with tetanus, Clostridium tetani. That bacteria, it grows anaerobically, it grows out in that soil. Okay, it grows in the soil. However, once it's exposed to the oxygen, it turns into this spore because now there's oxygen around. So it just exists as this spore. And they become airborne, they're all around us, right? What if one of those spores lands on the very sharp end of this paperclip, and now there's a spore here? And now all of a sudden, this gets driven into my hand, where there's no oxygen deep down in these tissues. That spore is like, oh, there's no oxygen. Guess what I'm going to do? I'm going to turn back into that active bacteria, and I'm going to start an infection, tetanus infection. Okay, so these spores are for survival. Anthrax, if you're looking at the anthrax scare after 9/11, okay, so they were taking anthrax spores, which are very difficult to destroy, and they can exist for long periods of time, mixing them up into some type of powder and distributing them. Okay, so someone would breathe in those spores, okay, or if it was cutaneous anthrax, into a cut, those spores would get into a cut, whichever method, and all of a sudden they were in the proper environment, they go back into the active bacteria and start an infection. So very, very scary these spores are. Not easy to kill. So if you're working with a patient with C. diff, and you're like, oh, okay, I just handle a patient with C. diff, there's spores around. I probably have, maybe I have some spores on my hand, I'm going to use this hand sanitizer. Hand sanitizer, okay, that's not going to kill the spores. They're too hard to kill. So what are you doing? You put the hand sanitizer in, on your hands. Well, you had a few spores on the palm of your hand, now you're just spreading them around. Okay, now I got them between my fingers, but they're still on your hands. They're still there. You didn't do anything by using that hand sanitizer. It does not kill spores. What do they tell you to do?
Wash your hands and then use hand sanitizer.
Wash your hands. That's the big thing. And then the hand sanitizer. Washing your hands, because what does washing your hands do? Where do the spores go? Down the drain. Okay, you're physically removing them from your hands. Are you killing them? Not necessarily, but you are physically removing them from your hands. A lot of people, you're looking over time, everyone getting a little bit lazier, just use some of this hand sanitizer. There's a lot of problems with that we'll talk about later when we spend some time talking about antiseptics and disinfectants. But things like that, nothing beats washing your hands, okay, to remove what is on them. So bottom line, with all of this, the word sterile is used when there is no life present. That includes a viral particle. That includes spores. Could I sterilize this stapler by rubbing it down with alcohol? Absolutely not, because I would not be able to kill certain bacteria, maybe certain viral particles, wouldn't be able to kill any spores if they were there. That's not a way to sterilize a stapler. I could toss it in an autoclave, so if you work in a healthcare setting, you have autoclaves present. Okay, that's what we have in the lab, so our lab tech will use to sterilize the media before he gives it to us. But that's what you would need. And after every lab we do, I'll sterilize, all the plates that had the bacteria on it so we can toss them in the garbage because you can't throw away live bacteria, of course. But sterile means everything is gone. Okay? Everything is destroyed. All right, this is our last gentleman that we'll talk about. So what happened was, there'd be women at the hospital giving birth, right? And these women would give birth in the hospital setting, and they would come down with an infection. And then there'd be some women at home giving birth, Okay, and we're talking, years and years ago where you're looking at someone's home just having like a dirt floor. Okay, so, but they would not be picking up the number of infections the women would in a hospital setting. You would think giving birth at home versus giving birth in a hospital setting, chance of infection will be much less in a hospital setting.